Combinatorial Effects of Metanosol and Radiation in Drosophila and Human Cancer Cells by Anthony Edwards, Mara Gladstone, Petros Yoon, David Rabin, Barbara Frederick, and Tintin Su. No single mode of therapy represents a cure for cancer. Multimodal therapy, in which more than one anti-cancer agent is applied in combination, has more promise and is being assessed in multiple clinical trials. The precise efficacy and degree of tumor control exhibited by combination therapies, however, remains variable. Although the reason for variability are unclear, discovery of additional novel drugs that synergize with an existing therapy, such as radiation, will allow multiple combinations to choose from, thereby increasing the likelihood of clinical success. How then can we identify drugs that work together with an existing agent to provide robust therapy against cancer? One solution would be to choose a suitable model and design screens that will allow co-discovery from the onset of an anti-cancer agent that's effective in combination, as opposed to screening for an agent that has an effect on its own and then testing whether it acts in combination with a second agent. Furthermore, a screening model that recapitulates the three-dimensional multicellular context of tumors might provide better predictive value than homogeneous single-cell culture models. Furthermore, a screening model that recapitulates the three-dimensional multicellular context of tumors might provide better predictive value than homogeneous cell culture models. We described previously a screen in Drosophila for chemical molecules that enhance the killing effect of ionizing radiation. Briefly, third instar larvae were irradiated and cultured in food supplemented with different molecules. Survival to adulthood was quantified 10 days later. Molecules that produced a percent survival that was 2 standard deviations lower than the average survival for the cohort were considered potential hits and were further tested for reproducible effect. A pilot screen through 1,990 molecule library using Drosophila checkpoint kinase 1 mutants yielded 4 molecules of interest. 2 of these are 20S-captothecin and topotecan, a derivative of camptothecin. Camptothecin and its derivatives inhibit topoisomerase I. Topotecan is an FDA-approved anti-cancer agent that acts as a radiation sensitizer of mammalian tumors. These results serve as proof of concept that Drosophila can be a useful screening model for anti-cancer agents that can be used in combination therapy with a standard agent like radiation. The pilot screen was performed on the Diversity Set library from the National Cancer Institute's Developmental Therapeutics Program. Molecules in this library were chosen for structural diversity rather than biological activity, and the library contains both natural products and synthetic molecules. Of the 1,990 molecules in this library, approximately 2% are from natural products, yet all 4 of the hits in the pilot screen were of natural origin. This led us to explore the idea that natural products would be a good source of molecules that increase the natural effect of radiation. Therefore, we next screened the Natural Products Set library from the NCI-DTP. This library consists of 235 molecules that were initially identified in extracts of plants or marine organisms. The active molecule in each extract was then identified, synthesized, and supplied in a 96-well plate format at 10 millimolar concentration in DMSO by the DTP. The screen was done blind, for example, without knowledge of the identity of the molecules in the whole library or in each well of the coated plate. The screen through the Natural Products Set, 235 molecules, using Drosophila checkpoint kinase 1 and p53 mutants, identified 13 molecules that reproducibly enhanced the effects of radiation. These included 3 microtubule depolymerizing agents, metanosine derivative, colchicine, and vincristine. Colchicine was the first drug we used in combination with radiation to design the Drosophila screen originally in 2006. Its identification, therefore, was reassuring. Vincristine is an FDA-approved chemotherapy drug against several cancer types and is being tested in combination therapy with radiation in clinical trials. It has also been used in experiments in Drosophila and was found to increase genetic instability in some assays. Metanosol isobutyrate, hereafter referred to as metanosol, was one of 4 hits in the pilot screen through the diversity set. Its repeat identification in the natural product screen attests to the robustness of the screening protocol. The effect of metanosol and its derivatives in Drosophila has not been described before and forms the focus of the article. Maytansine is an ansamycin antibiotic found in the Ethiopian shrub Maytanis. Maytansine inhibits microtubule assembly and induces microtubule disassembly in vitro, in cultured mammalian cells, and in embryos of sea urchins and clams. Maytansine exhibits cytotoxicity against many tumor cell lines and inhibits tumor growth in vivo. Early clinical trials failed owing to toxicity at therapeutic levels. Currently, there are multiple lines of effort to bypass toxicity by targeting cancer cells by metanosoide conjugated homing antibodies. Here we present a study of metanosol in conjunction with irradiation in Drosophila larvae and human cell culture. The results support the idea that Drosophila larvae can be used to identify molecules that have efficacy in mammalian models of cancer. Furthermore, we report differences in the behavior of Drosophila cells and Drosophila larvae that illustrate the importance of studying cell behavior in a multicellular context in vivo. Wild-type flies were of the Sevlin stock. P535A1-4 results from targeted deletion of the gene. Irradiation. Feeding stage 3rd instar larvae were irradiated as previously described. Briefly, 120-hour-old larvae were rinsed to remove food and passed through sizing sieves to obtain animals of uniform size. Larvae were placed in a petri dish and irradiated using a Torax X-ray generator set at 115 kilovolts and 5 milliamps, producing 2.4 rads per second. Irradiated larvae were then cultured on cornmeal agar media containing drug or DMSO carrier. Cell growth assay. The growth inhibitory effects of metanosol with radiation were evaluated using a modified tetrazolium salt MTT assay. In the MTT assay, 1,000 to 2,000 viable cells were plated in 100 microliters of growth medium in 96 well plates. Following an overnight incubation, metanosol was added at varying concentrations and the plates were irradiated on the same day, co-treatment, or 24 hours later, pre-treatment, and incubated for 6 to 7 days. The tetrazolium salt was added at a concentration of 0.4 mg per ml to each well following the 6 to 7 day treatment. The plates were incubated with the salt for 4 hours at 37 degrees Celsius. At 4 hours, the medium was aspirated off, leaving the dark blue formazan product at the bottom of the wells. The reduced MTT product was solubilized by adding 100 microliters of 0.2 normal HCl and 75% isopropanol, 23% milliQ water to each well. Thorough mixing was done using a TiterTech multi-channel pipetteman, and the absorbency of each well was measured using an automated plate reader. All experiments were performed in triplicate. Sensitivity of Drosophila larvae to metanosol and irradiation. Feeding stage 3rd instar larvae were placed in new food containing different concentrations of metanosol. Survival was calculated as the fraction of pupae that closed as adults, empty pupil cases over total, and normalized to survival of solvent-treated controls for each respective genotype. Error bars extended beyond fractional survival of 1 for solvent controls, 0 millimolar drug, simply because of the normalization. The data shown are averages from over 2,500 pupae per genotype, examined in 4 wild-type or 5 P53 different experiments. Feeding stage 3rd instar larvae were irradiated with doses of X-ray, as shown, and placed in new food containing different concentrations of metanosol. The dose of X-rays was chosen to achieve approximately 50% killing for each genotype. Most, if not all, irradiated larvae formed pupae in these experiments. Survival was calculated as a fraction of the pupae that eclose as adults from the pupal cases. The data shown are from one set of experiments in which data from 5 biological replicates were averaged per genotype per treatment. These data sets were 2 of several used to compute averages shown in Panel A, which are higher than those in particular experiments, but still within one standard deviation. Please note that p53 is a tumor suppressor gene. Metanosol inhibits the growth of HCT116 human colon cancer cells. Isogenic p53 wild type and mutant HCT116 cells were seeded in 96 well plates, allowed to grow for 24 hours, and treated with drug at the final concentration shown. Viable cells were quantified 6 days later using the MTT assay. The difference between wild type and mutant was statistically significant at the concentrations indicated with an asterisk. The data are from 4 different experiments. Please note that HCT116 human colon cancer cells have a mutation in p53. Irradiation of Drosophila larvae induces cell death in the diploid cells of imaginal discs. These cells are the precursors of adult organs. Successful production of an adult fly from a larva requires proper growth and differentiation of diploid imaginal discs. Following irradiation and induced cell death, remaining cells undergo compensatory proliferation to regenerate imaginal discs. Chemical molecules that interfere with compensatory proliferation and regeneration are expected to reduce the fraction of irradiated larvae that survive to reach adulthood. Microtubule depolymerizing agents such as colchicine, vincristine, and maytansine would disrupt mitotic progression and thus interfere with compensatory cell proliferation. We speculate that this is the basis for their ability to act in combination with radiation to kill Drosophila larvae. This would explain why 2 independent screens we have performed for chemical molecules that enhance the killing effect of radiation in Drosophila yielded 3 microtubule poisons from a total of 2,225 molecules. We acknowledge, however, that we cannot rule out the possibility that it is some other activity of this drug rather than the microtubule depolymerization that increases the radiation sensitivity. Drosophila larvae in human cancer cells behave similarly in the following ways. 1. Metanosol is more effective at killing p53 wild types than p53 mutant cells in Drosophila imaginal discs, and is more effective on p53 wild types than p53 mutant human HCT116 cancer cells at certain concentrations. In both Drosophila larvae and human cancer cells, metanosol generally increased the effectiveness of radiation. The extent of this increase varied, however, reflecting a mostly additive interaction between drug and radiation in Drosophila larvae, and additive to antagonistic interactions in various cancer cell lines. In addition to these similarities, Drosophila offers another dimension of experimental analysis, namely the ability to compare cells and multicellular tissues and/or organisms. For example, Drosophila p53 mutants, like human p53 mutant HCT116 cells, were more resistant to metanosol. In the context of a whole organism, this situation seems to be reversed. The p53 deficient larvae are more sensitive to metanosol than were the p53 wild-type larvae. This provides a clear example of how cells and multicellular organisms can behave differently and illustrates the value of studying drug effects in vivo in metazoan models. In conclusion, these results support the idea that Drosophila can be used to identify anti-cancer drugs. Furthermore, Drosophila offers an opportunity to compare the effects of small molecules on cells and on multicellular structures. We report here the results of a screen to identify small molecules that increase the effect of radiation. We are adapting the screen to identify small molecules that increase the effect of other standard therapies. Drosophila models of neurodegeneration are currently being used for drug identification or testing, both in academic laboratories and in industry. We hope that Drosophila models of human cancer will likewise become useful for identifying new anti-cancer drugs in the future.